So what this process is showing us is going to take us through step by step the the idea of the scientific method. So, what is the scientific method? It's a way to solve a problem. Scientists use this to understand what's going on here. Let's go through. This is a good overview of how scientists think and how I want you to be thinking when we're in class solving a problem. So, we're going to come up with the first thing that we come up with. We're going to ask a question. We're going to look at something and say, why? And that's going to be really important. This why is going to become this why or how or what is going to become our research question, and I'll refer to it as RQ in class a lot. All right. So when we ask that question, we then go and then we move in the direction of a little bit of background research. A lot of times, this right here, you're asking questions about something you know a little bit about, and then something kind of weird happens, and you say, hey, I wonder why that happened. So then we move into this next area, into the hypothesis. We're going to get into that in a little bit here. It's really important. That's just going to be our prediction, all right? So we're going to have to predict what we think is going to happen. From here, we're going to go ahead and we kind of like see a couple of cycles start to unfold. Typically what we do after our hypothesis, we're going to test it with an experiment. We're going to design an experiment and move this way. When we design the experiment, we execute and implement that. We're going to look at our procedure, and if the procedure is working, yes, that's good. Then we have some data that is helpful in drawing conclusions, and then we can look in a couple of ways here. We can go and say, all right, yeah, the results align partially or not at all with the hypothesis, because that can happen. It just doesn't fit with what we thought was going to happen, our prediction, or the results do align with the hypothesis. All right. No matter what happens here, you really can communicate results. So some people will tell you that if your results do not align, you don't need to communicate them. In class, we can end up going in either of these paths here. The two that are floating off to the side here, we need to look at and say, well, what are those there? Well, those are going to be some cycles in design. What's going to happen is if the experimental data becomes new background research for future projects to ask a new question, form a new hypothesis and experiment again. So what happens is if we get this data, a lot of times, and if you have enough time, we move in this direction. If the results don't align or they align partially, there may be some new question here. And that's awesome. This is really good science here, because the new question is going to allow us to further our depth of study and dive deeper and to a better understanding of our issue. So we're going to come back up here and make a new prediction again. Now, if the results align with the hypothesis, that's great. It makes sense, and that's a nice, perfect experiment. But science is messy and doesn't always work that way. Here we go back to here. If the procedure is not working, the answer is no. Then we need to go. We can't even get good data or get data, period. We need to troubleshoot our procedure carefully and set up, and then come back up this way and then test with a new experiment. So we need to continue to do that part as well. So we have a little bit of a cycle going here, and then we also get a little bit of a cycle going up there. So this is definitely the important part of the scientific method, and we're going to really dive into this in class a little further. So the process that we're looking at, there's a problem. We'll refer to it commonly, and I like to call it just a research question in class, RQ. So just remember, you're going to see that. You should know what that is. What are we researching? Our hypothesis is then what do we think is going to happen. So we talked about that being a prediction. And in our experiment, we're going to design a procedure for the experiment, implement that. Observations is data. Procedure, this is going to be where our data is collected. And then finally, conclusion, why did this happen? So there'll be an evaluation component there as well. But FEOC is the main part at PHEOC. Remember that. You can remember those 5 major parts of an experiment. Going into each individually, and I'm just going to go through these rather quickly because you'll have an outline to guide you through these parts when you're writing a lab report. But our research question looks at what's wrong, what are you interested in studying. It includes independent and dependent variables, and it always states things as a question. All right, so that's going to be important. Question, phrase it as a question. Hypothesis, prediction for your research, also referred to as an educated guess. We're going to phrase them as if, then, because statements. So you'll start hearing more about that if, then, because. It just makes it easier to structure. You, by all means, do not have to do it, but I suggest it, especially if this is new to you. The experiment, physical steps you take to test your hypothesis, collect your data, answer your research questions. This is written as a numbered list. This is the one thing that a lot of people forget is to number this thing. So these all should be numbered 1 through whatever. All right. This must be repeatable and testable. So somebody should be able to read your procedure and follow it. Observations, couple of vocabulary terms here that you should have down for sure. It's going to be quantitative and qualitative. So when we have our data that's collected, you're going to get one of 2 types of data, and I'm going to star this one because I'm just going to go out and say it that this is better. All right, this is better. This is the kind of stuff that we want to be able to look at, analyze, and evaluate, and that's what we need to do. So quantitative data is what your experiment should strive to do. This is numerical data. The plant grew 5.6 centimeters over 9 days. Those are kind of things that are indisputable as long as your procedure was designed pretty well, and they provide good data to analyze. Our qualitative data is still data that tells us something. There's no numbers in it. It's observational data, indicator solutions turning from blue to yellow. We do see shifts in color, but we still need to be able to explain what that is. Why did it change from blue to yellow? What happened in our solution? The issue there is it's a little too subjective, and what we end up happening is is that yellow or yellow green, or how yellow is it? And that's something that makes a lot of experiments, especially lab reports, a little bit watered down. Conclusions, we want to state if our hypothesis was supported or not and why. And we also want to talk about our data and how we can improve the experiment. And that'll be more so in the next part, actually. The data here is key. If you do not use your data in this section, you'll never receive full credit. All right, it is very, very important that there are numbers in there. I can tell you right now, when I read a conclusion, I can skim it for 2 seconds, and if I don't see any numbers written in that section, you haven't reached conclusions appropriately and back them up with the data that you work so hard for, by the way. Writing a hypothesis, if then because statement can be tested, and it needs to be specific. So we need to have all these three things in here. I'll give you some examples, and we'll go over this in class in a lot more detail. A research question here. Will salt and soil affect plant growth? That's an okay research question. We can probably improve it. Let's leave it for now. Here's an incorrect way to do this. Salt and soil may affect plant growth. So you see this word right here. I better use red for this because red's not good. May. No way. Don't ever use that. That's wishy-washy. That's not definitive. We can't allow that. All right? We have to make a firm prediction. That's what a hypothesis is. So let's see what a corrected version looks like. Here's your if, then, because, all underlined. All right. If 5 grams of salt is added to a tomato plant once a week, that's very specific. That's testable. Then we make our prediction. It will grow up smaller than a plant not exposed to salt. Okay, so there's going to be stunted growth here. And then the because, this should be an informed guess if you do your background research, but this can be any reason here why you think this is. Just provide one, your best guess. All right. If you did background research, it should be pretty intelligent. If you didn't do background research, it might be kind of off the wall. But no matter what you do, you need to have a reason here. We'll also work on identifying variables in our labs. So we're looking at control variables, independent and dependent variables. Control variables are conditions that are kept the same throughout the experiment. Things that you're making a point not to change. Things that are staying the same. So that's really key. So when you read about an experimental procedure, we want to find out what stays the same. That's going to be a control. The independent variable, this is the thing that I change. All right. So what are you trying to test? What are you doing in your experiment? It's something you're changing. You're altering in the experiment. You only have one independent variable for any of the experiments that we're doing in class. All right, so just one of these makes it easier to analyze and understand, and it allows us to keep our controls. When you're writing a lab report, include units, and your independent variable is usually on the x-axis. So when we graph this, and we'll talk about that, that's important for graphing. It's also an easy cheat, so you understand that when you're graphing, you don't have to think about it too much. Dependent variable is the last one. Variable you are measuring. That's the key here. Measuring. What are you recording? What kind of data are you collecting? Whatever you're collecting data of, that thing is probably your dependent variable. Something you're measuring, you only have one. Must include units, y-axis. This is a cool picture here. Something to help you keep them straight. I know I used to forget this stuff when I first learned it. Independent, what I change, the I in independent. Dependent variable, what you observe. And controlled is what you keep the same. We'll talk a little bit about units. When you're writing a lab report or collecting data, this is something you need to keep in mind. The base units that we need to do are going to be the meter, the gram, the liter, or the second. So whatever kind of data you're collecting, whether it's for length of something, the mass or the weight, right, the volume, all of those things need to be measured in these units. If we don't use them daily for the most part, are used to using this junk over here. We do not use the pint, the inch, the pound, the gallon, the foot. You're going to want to use that stuff. Make a point to use base units when you're measuring things. We'll work on that in class. When you do collect your data, your observations, I'm always going to ask you to provide some visual for that, and that's going to involve some graphing. When you graph, you need a lot of components here. It's basic, but I'm just reminding you need a title, y versus x, so whatever is on your y versus the x. Some explanation here. There needs to be detail of what's going on. Logical scale, all right, that's labeled, and the axes need a label with a measurement. So x-axis, say what it is, and then whatever the unit is that you measured here. So we need the unit, right? The unit needs to be here and here. So keep that in mind as you're graphing. The other biggest thing that we really need to focus on is not all graphs are appropriate all the time. So it's not like you pick a line graph because you like line graphs or a bar graph because you like bar graphs. So the line graph is show the relationship between two variables and shows data that is continuous. All right, this is key. Continuous, what does that mean? It's a logical flow from a start to a finish. So what do we see in this graph here? We go from 0, and the data is continuous, all the way down to 40 meters of depth. All right, they're not distinct separate groups. It flows that way. So depth in this case is continuous. The other thing that's continuous is time. If you have time down in the x for anything, right, that's continuous, you should have a line graph there. So keep that in mind. Bar graph used to compare data about two different organisms or things. Can be used to show data that is not continuous. Non-continuous data is important. It's not continuous. These are distinct groups usually, and groups is the first thing that I would look for if you have a bar graph that's appropriate. So in this case, they have these different species of bacteria. Down here, they're all distinct separate groups. There's no logical flow from start to finish here. There's no way to put it so that a line graph would be appropriate in this case. Procedures, just some basic things when you're writing your procedures out. Don't write in the first person with I, we. Don't do any of the he, she stuff either. Number each step. That's important. And include all the steps necessary in getting data. Is that a good procedure? Look at that. Oh, of course not. It looks terrible. This just isn't very repeatable. It doesn't tell me what to do. This is exceptionally bad. Observe your worm's behavior. How long, where, right? All those questions still remain. We want to be able to do the experiment the same as you, so you have to make it very clear and spelled out. The evaluation section is something we'll spend a little bit of time talking about in class, but describing sources of potential error and inaccuracy. We want to look at where in your experiment may some error have occurred, and then how would you correct this? This is important. You need to comment on those, one after the other. Here's where the mistake could have been made, and here's how it could be differently. Reflect on future research questions as well in that design cycle we did at the beginning. How would you do this differently the next time? In conclusions, we want to restate our original hypothesis. That's simple to do. Give specific examples. Talking about data again. Data. What do we say? We need that. Fully answer the research question. Do they support or negate the hypothesis? And then ultimately fully supported by data. Wow, that must be really important. Maybe I better write data one more time here.